Edvotech Instructional Videos presents Experiment 303, Exploring Biotechnology with Green Fluorescent Protein. This video will outline the pre-lab preparations for Modules 1 through 4. Experiment 303 has been updated to utilize the Enhanced Transformation Protocol, a new lysis buffer, and an improved staining method. Preparing the bacterial culture. Using one of the large LB plates you poured during the prior pre-lab preparations, create a source plate of the E. coli to use to create a liquid culture for the Enhanced Transformation Protocol. Do this by using a sterile inoculation loop to remove one of the E. coli GFP chromo-host bacto beads from the vial and place it near the edge of one of the plain, no-stripe agar plates. Using a fresh micropipette and tip, add 10 microliters of recovery broth to the bead to dissolve it. Use the inoculation loop to streak out the dissolved bead using the quadrant streak method. Click the link on the screen to visit the video for streaking plates. Label the plate, then place it in a 37 degrees Celsius incubator overnight, 18 to 22 hours, to grow the colonies. Make sure to invert the plate to avoid condensation. The control GFP plates can be made this same way, but using one of the double-striped LB plus AMP plus IPTG plates and the PT GFP bacto beads. Preparing the E. coli starter culture. Set a water bath to 37 degrees Celsius. Add 30 milliliters of recovery broth to the 50 milliliter conical tube. Using a sterile inoculation loop, swipe through a dense patch of E. coli, resulting in a clump roughly the size of a match head. Resuspend this clump of bacteria in the recovery broth by twisting the loop vigorously until it has released into the recovery broth. Shake or vortex the tube to fully resuspend the bacteria. Incubate the culture for 60 minutes in the 37 degrees Celsius water bath. After the incubation period, label 20 snap-top tubes E. coli. Swirl the tube to mix and add 1 milliliter of this culture to each tube. Place tubes on ice until they are needed for the experiment. Each student group will need 2 tubes of E. coli culture. Pre-lab preparations for Module 1. Label 10 snap-top tubes CSS. This stands for Competent Cell Solution. Dispense 300 microliters into each tube, cap, and store on ice. Dispense 500 microliters of calcium chloride to 10 snap-top tubes labeled calcium chloride and place on ice. Dispense 0.5 milliliters of recovery broth to 10 snap-top tubes labeled RB and keep at room temperature. Allow the PGFP plasmid, component D, to thaw on ice. Once it has thawed, dispense 12 microliters to 10 snap-top tubes labeled PGFP and place them back on ice. Since this is a small volume, make sure to tap the tube containing the PGFP plasmid or briefly centrifuge it to collect the contents at the bottom of the tube before aliquoting. To complete Module 1, your student groups will all need the following items: 2 tubes of E. coli cells, 1 tube of calcium chloride, 1 tube of competent cell solution, 1 tube of recovery broth, 1 tube of PGFP plasmid DNA, 2 Petri plates with a single stripe, 1 Petri plate with 2 stripes, and 1 unstriped plate. These plates were prepared in the Pouring LB Plates video linked on the screen. They'll also need 4 sterile pipettes and 3 inoculation loops. Make sure that your students have access to both a 37 degrees Celsius and 42 degrees Celsius water bath, a 37 degrees Celsius incubator, and ice baths or buckets, as these items are all needed to complete the transformation activity.
Preparation of the lysis buffer. Step 1. To make the addition of the lysozyme easier, add 500 microliters of the TEG buffer, component F, to the tube containing the lysozyme, component E. Pipette up and down to resuspend, then add this suspension back to the bottle of TEG buffer. Doing this makes addition of small masses much easier. Step 2. Add the entire contents of the now resuspended lysozyme to the bottle of TEG buffer and swirl to mix. Label the solution as lysis buffer. Step 3. Dispense 500 microliters of lysis buffer for each group. Refrigerate until needed. Module 3. Preparation of the column elution buffer. Step 1. Dilute the column elution buffer by mixing 35 ml of the 10x column elution buffer, component G, with 315 ml of distilled water. Step 2. Save 70 ml of the diluted column elution buffer for the preparation of the molecular sieve matrix, the next step, and dispense the remaining buffer for 10 student groups, 25 ml per group. Label this buffer 1x column elution buffer. Store on ice until needed, or freeze for up to 1 week. Preparation of the molecular sieve matrix. Step 3. Add 70 ml of the 1x column elution buffer to the bottle containing the dry molecular sieve matrix, component H. Step 4. Gently swirl or stir the bottle of matrix to ensure there are no clumps. Incubate for 30 to 60 minutes, stirring occasionally to fully rehydrate. Step 5. Aliquot 6 ml for each of the 10 groups. Make sure to mix the diluted, resuspended molecular sieve matrix between each aliquot to ensure even distribution of the material. Note, the prepared molecular sieve matrix can be used immediately or stored covered at 4 degrees Celsius for up to 72 hours. Control cell extract containing GFP. The control extract is provided for use by students that may not have positive results from Module 1 or Module 2. Step 6. Thaw the frozen control cell extract containing GFP, component I, at room temperature and immediately place on ice. Step 7. Label the tubes with GFP extract. Aliquot 220 microliters of the extract into the tubes. Place immediately back on the ice. Use this only in Module 3 if a student group's extraction has failed. For Module 3, each group will need the following materials: 2 screw-top microcentrifuge tubes, 1 chromatography column, 1 ring stand with a clamp, 1 microtiter strip, 6 milliliters of molecular sieve matrix, 25 milliliters of 1x elution buffer, micropipettes and tips, 220 microliters of controlled GFP extract if needed.
Reconstitution of lyophilized protein molecular weight standards and protein solutions. Step 1. Add 130 microliters of distilled or deionized water to the tube of protein molecular weight standards, component J, and allow the material to hydrate for several minutes. Vortex or mix vigorously. Step 2. The markers can be aliquoted for each pair of student groups, or students can share the rehydrated sample stock tube. The volume of sample to load per well is 20 microliters. Step 3. Store any unused portion of reconstituted sample at -20 degrees Celsius. Step 4. The 10x gel loading solution will be used for the native protein solution. Step 5. Label 10 microcentrifuge tubes as NPS and aliquot 30 microliters to each tube.
Step 6. Dispense 50 microliters of protein denaturing solution, component K, into 10 microcentrifuge tubes. Label the tubes as denaturing. Step 7. Preheat a water bath to 99 degrees Celsius or bring a beaker of water to a boil before the lab period. Preparation of electrophoresis buffer. Prepare the electrophoresis buffer by adding and mixing 1 part of tris-glycine SDS electrophoresis buffer, 10x, to 9 parts of distilled or deionized water. Example dilution calculations are provided. These are based on the number of protein electrophoresis units you are planning to use. Preparation for staining gels. Step 1. Prepare a stock solution of white vinegar and ethanol by combining 400 milliliters of white vinegar with 200 milliliters of ethanol. Gently mix and label as staining, destaining solution. Step 2. Add 125 milliliters of the staining, destaining solution to the bottle of flash blue protein stain. Shake briefly to mix. Step 3. Store both solutions at room temperature until needed. Step 4. Student groups will share 30 milliliters of flash blue protein stain, 140 milliliters of staining, destaining solution, water, a staining tray, and plastic wrap. Please note, white vinegar, sometimes called distilled or spirit vinegar, is an easy-to-find cooking and cleaning vinegar with an acetic acid concentration between 5 to 8% and a pH of approximately 2.6. Ethanol is a common laboratory alcohol which is available at various concentrations. Our flash blue protein stain has been designed to work with a wide range of white vinegars. However, we do recommend using 95% ethanol or higher. But if you have some old, unused Instastain protein cards on hand and you'd like to use them, the instructions for those can be found in the appendix on page 56. For Module 4, each student group will need access to the following materials: native protein solution, protein denaturing solution, 99 degrees Celsius water bath or beaker of boiling water with foil, the protein standard marker, 1x electrophoresis buffer, a polyacrylamide gel, 1 vertical electrophoresis unit, pipette and tips, staining tray, plastic wrap, spatula, flash blue protein stain, staining, destaining solution, and water.